Hello everybody, this is Peter Cooper from the NCBI. Welcome to today's webinar. This is NCBI Resources for NextGen Sequencing. So let's go ahead and start. Our speaker today is Valerie Schneider, who's in charge of the sequence offerings here at NCBI. And also in the room is Tom Madden, who is the BLAST lead at NCBI, and he's here to answer questions about Magic BLAST if any of them arise. I'm going to turn it over to Valerie now.
Thanks, Peter. And I want to welcome everyone to this afternoon's webinar. As Peter said, my name is Valerie Schneider, and I'm a staff scientist here at NCBI, who leads various teams that are building tools for sequence visualization and analysis. So today, in this presentation, I'm going to be introducing you to a few tools that are useful for analyzing and visualizing some next-gen sequencing datasets. And these tools include software such as Magic BLAST, which is used for read alignment, as well as the Genome Data Viewer, also known as GDB, which is NCBI's Genome Browser, and it can be used to visualize alignments in the context of other data that's mapped on the genome. And I'll point out that visualizing aligned next-gen sequence in a browser can be really a powerful tool for ensuring a good analysis, and that's a point I'll come back to a little bit later in some of the slides. I'm also going to show you how both of these tools integrate with the SRA, which is the NCBI repository for next-gen sequencing data. I'm going to be using these slides today that Peter mentioned that you can download from the webinar to either follow along online now, or you can follow along again at a later time. And I'll just point out that one of the easiest ways to get to the two main resources I'll be telling you about today is simply to Google the terms either Magic BLAST or NCBI GDB, and that'll get you to the tools that we'll be looking at. So the first tool that I want to introduce today is Magic BLAST, which is an aligner that was developed here at NCBI. You can find the code at GitHub. There's also a nice YouTube video that explains how to use it. I'm not going to really go into the details of the algorithm due to our limited time today, but it's been recently published. So for those of you who really want all the gory details, you can find that here in the publication. And down here at the bottom is the PubMed ID, so you can look that up. But in short, what it does is it couples aspects of the previously published Magic Aligner with BLAST database infrastructure. What it's doing is it's checking for overrepresented target sequence fragments during its seed selection, and it uses an innovative greedy alignment extension procedure as it runs. Now, Magic BLAST can produce splice alignments when it's aligning RNA on the genome, and it selects the best matching position genome-wide by optimizing a spliced alignment score for either single or paired reads. So that's just a really high-level overview of what this tool does. But with all the aligners that are out there, why in the world would you want to try Magic BLAST? Well, first off, as shown on this slide, it's really quite versatile. It runs with both RNA and DNASeq runs. Again, you can align against a genome or a transcriptome. It's suitable for using either long or short reads. It's both splice and pair aware, and as I'll be showing you in a little bit, it's got this really cool ability to automatically retrieve SRA data so you don't need to download it. And the tool itself excels in the following areas, whether it comes to finding introns, handling high levels of mismatch, handling compositionally biased DNA, or even its ability to handle these long read RNAseq data. And this is all compared to other aligners that you might be familiar with, like HiSet2, STAR, and TopHat. And a more detailed explanation of those comparisons is available in the publication. So the other tool I'd like to introduce today and get you oriented with before we start digging into the data is the Genome Browser, or GDB. So in this view here, I have highlighted the graphical display, which we'll be configuring a little bit later with this tracks button here to show tracks of interest. Some other browser features that we'll be looking at today are this pick assembly tool that's up here at the top left, as GDB supports multiple human assemblies. And in fact, if you work on things other than human, you'll see that you can also open GDB instances for different organisms as well through this little tool. We'll also be looking at the search option so that we can navigate to a region of interest. We'll take a look at the user data and track hub section, which is where you'll be adding externally sourced data into the display. and also this add tracks section down here at the bottom, where you can directly enter NCBI accessions for GEO, SRA, and dbGaP GWAS data that's already been mapped to the genome, again, also that you can see this data right in the graphical display. So in order to take a look at the utility of tools like Magic Blast and GDB, let's take a look at this following hypothetical case study. So we can imagine that we've got a collaborator who's studying Charcot-Marie tooth disease. And for those of you who aren't familiar with CMT, it's a hereditary neuropathy that affects about 1 in 2,500 people. It can be caused by mutations in several different genes. There's at least 5 sort of well-characterized ones and a growing number of sort of less common ones. But it has dominant, recessive, and X-linked forms of transmission. So it's a pretty common neurological disorder, and you've got this collaborator studying it, and they've asked you to realign some RNA-seq reads that were associated with a publication that they were reading, in which these inducible pluripotent stem cells were used to study this disease. And they want you to do this analysis for them because they really want to take a deeper dive into the data before they decide to undertake a similar analysis themselves. So this might be the type of request that you might get. Your collaborator is also going to ask you to help them with a second analysis in which they're now going to have you align some DNASeq reads, now not RNAseq, but DNASeq, from a different Charcot-Marie tooth-related publication. And they'd like to get these reads aligned to the latest version of the Human Reference Assembly, GRC-H38, because they specifically want to evaluate the way the alignments look and the coverage ratio at a particular location, this SNP, RS78993751, which is a site in the publication which they had read. The authors had proposed that perhaps there was some mismapping of the read data in GRC-H37. So today, we'll take a look at the NCBI tools you can use to help your collaborator with this, and also maybe take a look at what some of the conclusions that we can draw from the types of analyses that we'll be doing. So to get started on either of these analyses, the first thing that you're going to need to do is generate the necessary alignments. And as I'm going to show you here, this takes just a few steps. And in this case, we need to start by defining the data sets from SRA that we want to align. And fortunately, because we've got our collaborator here, they're just going to tell us what these are. But otherwise, if this were your own experiment, you could go looking for your data sets of interest, either in publications or maybe using some of the SRA tools like the Run Selector or the Run Browser to find sets of interest. But in this case, we've got a publication here with this PubMed ID, and they've identified 2 RNA-seq data sets right here, one which represents data from some experimental samples, and another one which represents RNA-seq gathered from the control samples. And just so you know, these 2 data sets are on the order of about 10 gigabases each, and they happen to be 150 base pair paired Illumina reads. For the DNASeq data set, again, they've told us the publication, and in that publication, we can find our SRA data set. And here, this is a much larger set. It's 148 gigabases, but again, we're looking at paired Illumina reads, in this case, 100 base pair runs. So this is the RNA-seq and the DNA-seq that we want to align. Our next step is to create a BLAST database and to generate the necessary alignments. And as I'm going to show you here, this really just takes a couple of quick and easy steps. The Magic Blast help documentation provides a really nice example that demonstrates how simple it is to set up the GRC-H38 BLAST database. So really, it's 3 lines of code right here, and you can just use this yourself, and you will get yourself a BLAST database set up. Now, generally, if you're going to run Magic Blast, you can do this with default parameters, but there are a few parameters that it's worth making mention of here because these were ones that you'll want to pay attention to. First off is this parameter for SRA. So if you are aligning data from SRA, you just plug in the accession right here. So this is the cool thing about Magic Blast that I want to reiterate here, is that you don't have to go to SRA and download these reads from SRA. For Magic Blast, you can just give the tool the SRA accession, and it'll take care of the rest for you. So that saves you a bunch of time and disk space in terms of working with this. Another couple parameters to know, if you're aligning either RNAseq or DNAseq, you want to pay attention to the paired parameter so that you can declare whether your reads are paired or not. And if you're aligning DNAseq, you also want to pay attention to the splicing option, because you'll want to disable that in the case of DNAseq because we're not splicing. Now, depending on your compute resources, you can also choose to specify whether you want multi-threading or not. So the default output for Magic Blast is SAM, which you can then convert to BAM files and index them using public tools like SAM tools. Now, for the data sets we were working with, the BAM files that got created for our RNAseq were on the order of about 2 gig apiece, and our DNAseq was about 28 gigs. So this is a pretty big data set. And again, you can access these output BAMs from the FTP site that's listed here on this slide. So if you want to play around with these and use them in the browser, as we'll be doing, that data is available to you. Now, if you are working with your BAM results and you want to visualize them in GDB, it's going to be important to make them publicly accessible. So that means putting them on an FTP site. And again, you can use the FTP site that's right here to do that for any follow-up that you want. Now I'll mention, if you're working instead with results from your aligner that you don't want to make public, so maybe you're not using a public SRA data set, you're working with your own data set, NCBI does also offer a desktop tool known as Genome Workbench, in which you can do pretty much all the same analyses and then some that I'm going to be showing you today in the browser. Today we're going to be looking at public data though. But the point I just wanted to make with this slide is that really getting your alignment run set up in Magic Blast, from everything from getting your reads to setting it up to getting your output, really is a very simple and straightforward thing to do. So now that we've gone ahead and generated our alignments, let's go ahead and get the Genome Browser, GDB, set up for our analyses. Now, if we already had GDB open, which remember you can find easily on the web by just Googling NCBI GDB, the first thing that we're going to want to make sure that we do is to select the GRC H38 assembly from the Pick Assembly menu here, since that's what we aligned our RNA-seq data to. And the next thing that we're going to do is use this Tracks button here to configure our display. And we're going to take advantage of a feature known as the NCBI track sets, which you can find under this menu here. And track sets are pre-selected collections of high-value tracks that are relevant to different types of analyses. And today we're going to be using the default track set, which contains, as you can see here, genes, a whole bunch of different SNP tracks, all SNPs and some other subsets of SNPs. And then the last thing that we're going to want to do is add our BAM results to the viewer. And we're going to do that by using this User Data and Track Hubs section. And from there, we're going to select the Add Remote Files option. We will enter here the URL where our BAMs are located. And then we can give these an optional name for the track. So in this case, I'm looking at my control data, and it's control SCs because these stem cells were induced into Schwann cells. So this is just a name to help me remember which data set that I'm looking at. And once I click the Add button here, this panel will change and it will tell me that I have added my track, and it gives me the name of that track right there. So we'd repeat this step a second time to add our other RNA-seq BAM file, not for the controls, but for the experimental data set. And that will load everything that we need into the browser for analysis of our RNA-seq. Let's go ahead now and get oriented to this display for GDB, which has defaulted to a zoomed out view of chromosome 1. So we can see the sequence across the top here, and below we have a bunch of different data tracks. Now I'm using this orange bracket here on the side to highlight the various tracks that came from that default track set that we used. And the purple bracket down here highlights the 2 BAM tracks that we added last. And I want you to notice this green highlight here in these 2 BAM tracks. That green highlight indicates that it's externally provided data. So this is the data you added to the browser. It's not the data that NCBI is showing you. So anytime you add data of your own into the browser, it'll get one of these green highlights on here. Now, in order to declutter this display, because I really don't need all of these tracks on here to do my analysis, I can remove some of these, and I can do that by clicking over here on the right-hand side for any particular track. We'll just zoom in, and I can click on this X button here to remove that track. So I'll be removing a few of these SNP tracks and expression tracks that we see here with the graph because I'm not going to need them for right now. But I also want to point out that I can reorder, and sorry, the tracks I'll remove are the ones that are highlighted here with the red bracket. And if I want to, I can also reorder the tracks in my display by simply clicking and dragging on a track that I want to move, and I can just pull it to its new location. So this display is very customizable for your analysis purposes. So if we move on now, we can see our cleaned up display here. Like I said, I got rid of a bunch of SNP tracks and some extra gene tracks that I didn't need. And you'll notice that at this very low zoom level, zoomed all the way out to see all of chromosome 1, the alignment track down here is showing as a coverage graph. We know it's a coverage graph. This is highlighted here with some text, and it's scaled at log 2. Now, a quick and dirty look at this display tells me that my RNA-seq data is pretty evenly aligned across the genome. I've got pretty good coverage across here, except for this big gap, which happens to correspond to a large gap in the sequence of chromosome 1. So as I mentioned earlier, this kind of eyeballing can be really helpful when you're doing RNA-seq as an early step of QC, because you've run your wet lab experiments, you've got some data. And before you start delving into all the deep analyses that you might want to do computationally, this just helps you show, did everything actually work out the way I expected? Did I get good coverage across the genome? Does it look nice and even? I can zoom into various levels and just look at features. And again, it's just a nice way of checking your experiment before you invest a whole lot of time in doing your analysis. And so we're going to move on now and take a look at some of our alignment data a little bit more closely. But first, I want to tell you a little bit more about this alignment display. If I were to click on this gear icon right here that you find at the far right, this is where I find the menu that I can set my alignment display. And what we're looking at here is that we can see that it is in something called adaptive mode, which means that the display will look a little bit different as I zoom to different sequence levels. And so as we'll come back to in just a little bit, we'll be using this menu to change the display, but we access it right through this gear setting right here. So now let's go ahead and zoom in a little bit. We're going to go back to that search box I mentioned earlier, and we're going to zoom to the location of a specific gene known as S100B. Why this gene? Well, our collaborator asked us to look at this gene, and it's because the gene happens to be involved in Schwann cell development. And again, we are looking at some inducible pluripotent stem cells that the publication induced to develop into Schwann cells. And the authors of this publication reported that this gene appeared to be differentially expressed between the controls and the experimental stem cells. And so again, we want to look at this a little bit more closely. So we're going to go to the location of that gene. And so now we have jumped to this new view. We are zoomed in a little bit up here at the top. Here I have just the S101B gene shown in green. And we're now at this mid-zoom level. And so we've lost our summary graph, so we're not seeing coverage or pile-up displays. But instead, we're seeing individual read alignments here. And at this mid-level zoom, though, we're not looking at the individual bases. We're just getting sort of a general sense of what the read alignments look like. If I cut this image down a little bit so that we can see a little bit more clearly what we're looking at again here, here's our gene. Down here are our alignments. If I then zoom in to the level of a specific exon, again, just to get a little bit closer look at what is this alignment display actually showing me. So if I look at the level of this exon, I can now see some features of my alignments. I can see mismatches with these little red tick marks. I can see intron spanning reads. So the gray part, the light gray is the aligned portion. This thin gray bar tells me that this read spans to another aligned portion that's in a different intron. And I've also got these glyphs here that show me that I have some unaligned tails in my reads. Now, this is still a little bit busier than I probably need in order to analyze the gene expression at this particular locus. As we saw when we were zoomed out, we had a pretty big long trail of alignments in the display. So I'm going to go ahead and use that settings cog to update the display for my alignments. And what I'm going to do is I'm going to set the display to packed. And by setting it to packed, that's going to force a coverage or a pile-up graph in my display. So once I do this, here's my pile-up again. Now I'm still zoomed at the level of the gene. And the results become strikingly obvious when I compare expression for my control data and my experimental data, with essentially no expression detected at all in the experimental track, because we can see there are no alignments here at all. So this confirms the results of the publication that my collaborator was interested in. I can see that this gene is differentially expressed in the control versus experimental conditions. And so I can pass this information on to the collaborator. I can show them this display so that they can assess it for themselves and really get a good sense of how different was the expression. In this case, it's really obvious, but they now have the information that they need to make the next decision about their own research plan. So at this point, we've taken care of the RNA-seq analysis that our collaborator wanted, and we can move on to the second analysis for the DNA-seq and compare the alignment profile in both GRC H37 and 38. And to do that, I'm going to move along by first just using this reset button to reset my GRC H38 display. And then I'm going to go back to this pick assembly tool, and I'm going to update my display so that I'm now looking at GRC H37. Once I'm in the GRC H37 display, once again, I'm going to go ahead and tailor it so it shows the tracks that are most helpful for the evaluation I want to do. And in this case, I'm going back to my track sets. Now I'm going to use my genetics and variation track. And then, as I showed you before, I can clean out tracks that I don't really feel like I want to look at. And I'm not going to go through all those steps here. You now know how to do that. Now, I know from my collaborator and the publication that he gave me that the alignments for this DNA-seq data to GRC H37 already exist. They were submitted to SRA along with the DNA-seq reads. And so I don't even need to run Magic Blast in this case to look at this data. In fact, all I have to do is use this Add Tracks tool down here on the left-hand side, and I can enter in my SRA accession. And so I do that, and the alignments will load automatically into the display, and we'll see those in just a second. Now, because my collaborator has told me that he's interested again in looking at the alignments at a specific location, I'm going to go ahead and I'm going to enter in the search box the coordinates on chromosome X that I want to go to, and I get my display here. Now, I'll just point out that, at this zoom level, if I were to mouse over my alignments, I can get a pretty nice looking tooltip with a lot of interesting and useful information about my alignment. It tells me the percentage identity, the coverage, mismatches. If I want to know the actual alignment itself, there's a cigar string down here. And if my read had any unaligned regions, by clicking on this little view button here, I could see these three unaligned bases that are at the 3' end of the read. So I just point this out that if you ever want specific details about a given read alignment, that's available to you at this very zoomed in level. Now, to better evaluate the coverage and the allele ratio at this base, which is what my collaborator wants me to do, I'm going to go ahead and use the following settings for my alignment display here. So again, going back to my settings menu, I'm doing the following thing. Even though I have my adaptive display on, I am clicking this box to show pile up. And so what this does is it forces the display of a pile up even at the highest zoom level, so that even though I now still have the read alignments, as we saw down here, as we'll see in the next slide, I will also have pile up information displayed alongside that. Now, I'm also using this feature here, this option for pile up display. So instead of looking at a graph like I did earlier, I'm going to actually have my pile up show as a table. And what it's going to show me is the percentage of reads that either match or don't match the reference sequence. And so once I set this, I can go back here and look, and I can go now again, my site of interest here is highlighted in green, and I can see that something is clearly amiss here. I can look at this table and I see that 48% of my reads don't match the reference base at this position. 51% do match. So that looks like my sample is heterozygous at this position. Why is this a problem? Well, the sample that I'm looking at here is a male, which means that they only have one copy of the X chromosome. So genetically, they shouldn't be having two different alleles at this location. They should be hemizygous. So the fact that I'm seeing two different bases represented at about a 50-50 distribution tells me that something is not quite right. So I decide that, my collaborator told me that the authors of the publication thought that maybe there was some mismapping going on at this location in 37. So I want to check this out in the newer version of the genome, build 38. So looking up at my SNP track that was in my display, I discover that there is a SNP at this location, so I can get my SNP ID right here. I can then update my GDB display to go to GRCH38, and I can search for this SNP location. So now I'm looking at a display of chromosome X in GRCH38, whereas in the previous slide we were looking at GRCH37. And I'm using the same track configuration, but now I need to go ahead and load my data from my DNASeq analysis I did with Magic Blast in. So again, now we have our green track down here. I'm zoomed in, I've got my alignment display, I've got my same pileup as I did before. I'm at the location that I wanted to be at. And if I zoom in, I notice that I no longer have any evidence of what appears to be heterozygosity at this position. And I also happen to notice from my table that I've got actually half as many reads aligning. So I've got 22 reads aligning here in build 38. And if I go back to 37, I had 43 reads aligning here. So something has clearly changed in my alignments of the same DNASeq data to the different versions of the assembly. So what's going on here? Well, if I dig in a little bit more closely again, one of the tracks that I have in my display here is this model-based paralogous sequence differences. And we see that there's a paralogous sequence difference that is located at exactly the position that my collaborator was interested in. So what does this mean? Well, in order to keep what could be a very long story short, I'll tell you the following, that subsequent analyses of the human reference assembly showed that prior versions of the reference were missing sequence from chromosome 2 that corresponds to a pseudogene that's related to a gene on chromosome X at the position our collaborator was interested in. And so looking at the read alignments in GRCH37, the unusual allele count and the higher density of read mapping that we saw was due to mismapping of reads. So reads that should have belonged to the sequence on chromosome 2 didn't have a home to go to, and they ended up aligning to chromosome X. That chromosome 2 copy had a different base at that position, so that's my paralogous sequence variant. And in fact, if we zoom out to a lower zoom level here, looking at GRCH37, we can see the evidence of these mismapped reads in a very localized region. We see the sequencing depth get higher, and we see the incidence of mismatches going up. So again, this is another one of those nice examples where having a visual display of your alignment data can help you understand and interpret it in ways that sometimes just looking at the output of running various bioinformatics programs can't. In GRCH38, the newer version of the assembly, this missing sequence on chromosome 2 was included in the assembly. Those reads, now when I ran my Magic Blast alignment, were able to find their proper home on chromosome 2. And so again, if I look at the zoomed out view, I see much more even distribution of reads across the region, and I no longer see the mismatching. So I can share this information with my collaborator, I can show them this graphical display, and they can now sort of better interpret the results of the original publication, and they can go on their way to their next experiment. So with that, we've come to the end of our case file and our analyses. As I said, you can use the slides that are available to you and the alignments to walk through this stuff at your own pace. What did we learn today? We learned how to use Magic Blast to align RNASeq and DNASeq directly from the SRA to two different versions of the human assembly. We used GDB to visualize RNASeq alignments, and we looked at their expression profiles. We also saw how we can load alignments that are in SRA already directly to GDB. And lastly, we use GDB to evaluate the coverage and allele profile at a very specific locus. So I hope you found this demonstration informative. And please let us know if you have additional questions about the browser or any data in the tracks. And I just have a couple slides here at the end that are just some shout out for some new information for NextGen data that's happening here at NCBI. The sequence read archive, we have started making data available in Google and Amazon cloud environments, and by the end of the year, we should have authorized access data available on both cloud platforms. Additionally, we're working on getting several other tools and workflows into the cloud, so that includes BLAST, public SRA data, and our prokaryotic annotation pipeline is also moving there. And lastly, NCBI is engaging in a bunch of different codeathons to help people learn about our new tools that are moving to the clouds and data there. So be on the lookout for announcements from NCBI about these tools, and we hope you'll join us in some of those. And so with that, I'd just like to thank everyone involved with Magic Blast and the browser teams. Thank you.
Thanks, Valerie. There's a couple of questions about content that I did not know how to answer that Valerie might be able to help with. And someone asked about filtering Magic Blast plams, and they mentioned MapQ flags or anything else, thinking about multi-mapping or discordant reads.
Within the viewer, if you're asking, we do have in the display options options to stratify your display by forward and reverse reads. We also have coloration for PCR duplicates. Anything that is very poorly mapped also shows up all in red. We have recently added the ability to haplotype sort reads within the viewer. So if you have like 10x data and you know that something is haplotype 1, haplotype 2, that can be sorted. Within Magic Blast, I believe at the moment when you just, and I'll have Tom maybe answer this, but when you take from SRA, I think you're just taking the full set of reads. If you wanted to subset that to only align reads that had a particular quality score on the mapping, I think you'd have to do that separately. When it comes to displaying reads that are just a given map quality or not, we don't have a setting in the browser for that.
Okay, another question that I think I know the answer to. Is it possible to run GDB locally, and how complicated is it to add a new species? I think the answer is it's not possible, but I think Genome Workbench might be able to solve the problem.
Yeah, GDB does not run locally. I will point out that if you, again, just Google NCBI GDB, you'll end up at the landing page. We have GDB instances for over 700 annotated RefSeq assemblies, so we support quite a large number of organisms. If you want to look at just a private genome assembly, again, Genome Workbench, our desktop tool, would certainly allow you to load all of that data in there and visualize it in a graphical display. And it's built on the same code, so the displays and the functionality feels very much the same as the web browser. So yeah, if you have your own genome, you can load it to Genome Workbench.
Valerie, could you, if you can you change to a web browser and show people where they could find out about Genome Workbench?
So this is our tool here. You can go to the download link here on the right-hand side, and we offer builds for a number of different operating systems. And as you'll see here, there's quite a rich collection of help documentation, both in the forms of YouTube videos and online tutorials for setting this up.
Another question. It's about the limit in the size of SRA data that you can load into the browser, I think.
No, so if you are loading remote files, we are streaming that data. We don't pick it all up and load it at once. So really, we support very, very large BAM files. You can just stream them over there because we're just loading what is in the display at any given time. And we're also doing some caching. So if you come back to a site that you've looked at already, it'll load up very quickly into the display. And for the data that's in SRA, I've yet to encounter an issue with trying to load up any SRA read alignment data into the viewer as well.
Okay, I think we'll do one more question, but I'm not sure I completely understand it, and maybe you will. The question is there an easy way to show regions which have overlap between multiple tracks and capture this information in the viewer? For example, I want to focus on genome regions which have significant overlapping regulatory features such as TFPs, DNAs, and code peaks.
We don't have a mechanism to find that sort of jump the browser to places where there is overlapping data in lots of different tracks. You can certainly load up any of the encode data. We support UCSC track hubs. And in fact, if I'll just bring this up here right now so you can see this. This section over here for user data and track hubs, if you click this Add Track Hubs button, you're able to search the track hub registry and find any track hub, so encode data is available through there. We also have an NCBI provided track on regulatory regions associated with genes that was developed by RefSeq. You can load all of these tracks up into the browser and sort of zoom out and pan along to find regions where you might find significant amounts of overlap in data between different tracks. But that's a really interesting suggestion that if we had a way to help jump the browser to places where for any given collection of tracks displayed, there was data in 2 or more tracks.
Okay, so that's all we have time for right now. I think we'll go ahead and end the webinar. If there are any additional questions that we didn't get to, I'll make sure that they're added to the Q&A document that's going to come out after the recording's available. So thanks, Valerie, and thanks everybody for coming, and I'm going to go ahead and end the webinar.